Our next speaker, Frank Jaksch. So I can say that Frank Jaksch is the co-founder of ChromaDex, one of the main suppliers of nicotinamide ribosides that are being used across the globe in a number of clinical trials. Frank, whenever you're ready, you can start we can see your slides and you can start the slideshow. Perfect. Looks great. Go ahead.
All right. Thanks, Morten. We really appreciate the invitation to speak today. Good morning, everyone from California. Another back-to-back. You get two guys from California, I guess, here. So I wish I could have been there in Copenhagen today. I was really trying to make the travel work, but there's one thing I love more than aging research and NAD precursors, and that's watching my 13-year-old son play ice hockey. And he has a tournament that starts here in California today. So yes, there is ice hockey in California. So today I'm going to focus my presentation on recent advances with nicotinamide riboside in orphan diseases and aging. I want to give you a little bit of history first. The ChromaDex journey with NR and NAD precursors started with Charlie Brenner's 2004 publication, Discoveries of Nicotinamide Riboside as a Nutrient and Conserved NRK Genes, establish a price handler independent route to NAD in fungi and humans, as well as his 2007 publication, NRK Structures Reveal New Pathways to NAD. Another important piece of the puzzle was a review by Khan et al. From Columbia in 2007. NAD metabolism is an attractive target for drug discovery. In 2008, we started evaluating the patent landscape and started down the path of licensing patents and trying to figure out a way to produce NR at commercial scale. And then it took us about 5 years or so, but ChromaDex launched NR as a branded ingredient called Niagen in 2013, which ultimately led to the launch of a consumer product that we call True Niagen in 2017. For launching Niagen in 2013, we've seen a tremendous amount of interest in the research community for NR as an NAD precursor, which led to the launch of what we call the ChromaDex External Research Program, or CERP. Since 2013, we have signed more than 235 collaborative agreements representing an estimated probably 85 million in total research funded through the program. CERP has also resulted in numerous patent applications and licenses as well. And additionally, to date, Niagen has been part of more than 65 publications, including 13 peer-reviewed clinical trial publications. These trials have demonstrated the safety and effectiveness in boosting or raising NAD, particularly in aging populations. There are multiple studies in the pipeline that are investigating NR's potential role in diseases related to mitochondrial dysfunction, aging, and what I'm going to talk about mostly today is about orphan diseases where there are research ongoing. So Niagen has been shown to be a safe precursor to NAD with demonstrated evidence of elevating NAD or increasing NAD metabolism. One of the ChromaDex sponsored studies, one of the ones that we funded, examined the safety and efficacy of escalating doses in Niagen in 132 healthy overweight adults. In this study, it was a double-blind, placebo-controlled parallel arm trial that took place over the course of 8 weeks. Participants were given 100, 300, and 1 gram of oral Niagen, or NR, or a placebo daily. On average, the study participants that were consuming the 300 milligram dose, which is kind of where we are in the market right now with our True Niagen product, showed a statistical significance of increase of about 51% in whole blood NAD. And that was over the first 2 weeks where we saw it sort of plateau. And it stayed and maintained that increase over the total of the 8 weeks of the study. There are over 40 active registered trials that are ongoing right now if you go to clinicaltrials.gov. So that's a pretty significant pipeline that we have. And hearing some of the question marks over the future regarding translation from mouse or from animal data into human data. We hope that some of these studies are going to at least go towards answering some of those open questions, because there's only a few that are out there right now, at least from our perspective, but the 13 that were out there, and we're learning from those and then trying to figure out what the causes of those might be, and hopefully we'll be able to solve for that in these future clinical studies. Some of the outcomes that we're looking at in these studies are muscle and locomotion, brain and cognition, kidney function, autoimmune disorders. And today I'm going to talk about or summarize some of the work that we're doing related to rare diseases. When we launched the CERP program in 2013, we didn't expect to see this much research interest in rare pediatric orphan diseases. And I'm not sure if any of you have had a chance to meet with or talk with parents of kids with these types of rare diseases. You know, they would essentially do anything for their kids. And while they're always looking for a cure, they're also looking for something that might improve the quality of life of their child's life while they're here. So for us, it's been exciting to see research on NR moving the direction of potentially helping or providing a potential solution to improve the quality of these kids' lives. You know, we don't look at this as something that from a cure perspective, it's more of, can this improve the quality of kids' life that are suffering from these types of diseases? So DNA damage and mitochondrial dysfunction are central themes of many of these orphan diseases or diseases of rapid aging. The root causes of several of these disorders can be traced back to these rare genetic mutations that impact DNA repair mechanisms, which can result in mitochondrial dysfunction, defective mitophagy, organ dysfunction, such as rapid neurodegeneration as an example. Depending on the disease, disrupted NAD metabolism can either play a role, a casual role as it's closely linked to DNA misrepair or mitochondrial dysfunction. As shown in this graphic, the NAD depletion from metabolic stress or defective metabolism impacts a number of age-related pathways. NAD plays a critical role in the function of neural tissue. Dysregulation of NAD metabolism is believed to contribute to the progression of age-related neurodegenerative diseases such as Alzheimer's, Parkinson's, Huntington's, and ALS, among a few there. We do have active CERP programs in all those categories right now. Also, in certain orphan diseases, this process of degeneration can be accelerated, appearing in impacted infants and children mostly. So we're looking at mainly pediatric orphan diseases. Since the launch of CERP in 2013, we've started doing quite a bit of research on orphan diseases such as Cockayne Syndrome, Ataxia Telangiectasia, Werner syndrome, Citrin Deficiency, Duchenne muscular dystrophy, Friedreich's ataxia, Leigh-Ferrand syndrome, and mitochondrial myopathy. Today, I only have time to go through the first 5 there, so I'm going to try to sort of give you a summary of what we've been doing on those first 5. So we'll first take a look at Cockayne syndrome. CS is a rare and inherited form of dwarfism that includes premature aging symptoms, such as wrinkling of the skin, thin nose, dental decay, loss of hearing, vision-related issues, and premature graying of the hair as well. Preclinical research has evaluated NR's role in CS. Our very own ARDD organizer here, Morton, and his colleagues found that NAD-NADH ratio was decreased in cells from CS mice in comparison to controls, and NAD supplementation rescued the CS-associated phenotypes in human cells and mice. 1 week of NR treatment in a CS mouse model normalized NAD and ATP levels, while improving membrane potential, decreasing ROS production, and normalizing cerebral transcriptome. And our supplementation also attenuated changes in the mitochondrial pathways and pathways associated with oxidative stress, transcription, DNA repair, DNA pair response, or DNA damage response, and histone acetylation in the CS model. In the figure to the right, you see the results from a preclinical study conducted at the National Institute of Aging. The study showed that oral NR administration reduced the age-related hearing loss and improved cochlear health in CS mouse models as early as 10 days in that study. Our team is hoping that these findings will support justification for future clinical studies in evaluating NR and CS patients. We have seen progress in other ones where they have transitioned to human, and I'll show you those as well. So if we move to AT, which is a hereditary progressive neurological condition characterized by difficulty in coordination of voluntary movements, small clusters of enlarged blood vessels in the eyes and on the surface of the skin, a weakened immune system, and increased forms of various cancer. Symptoms of AT include progressively impaired walking as early as age 5, neuropathy, slurred speech, difficulty with eye movements, reddish lesions, and increased risk of respiratory infections. AT is caused by mutations of the ATM gene, which encodes for the ATM kinase required for repair of double-stranded DNA breaks and DNA damage response signaling. Unfortunately, there's not an effective treatment or a cure for that matter for neurological manifestations of AT. Mitochondrial dysfunction and cellular senescence have been demonstrated in AT patient fibroblasts and in animal models of AT, similar to which is observed in aging. In the Yang et al. Study, increasing NAD with NR removed damaged mitochondria by stimulating mitophagy and preventing senescence and neurodegeneration. NR was also shown to improve mitochondrial homeostasis and motor behaviors. Translation of these findings has resulted in 2 registered clinical NR-related clinical trials in AT patients. The most recent study, which is being conducted in Norway with children with AT, will be treated with Niagen for 2 years and evaluated for improvements or maintenance of motor, mitochondrial, liver function, as well as glucose metabolism. Werner syndrome is a rare form of adult progeria, essentially, caused by mutations resulting in the loss of function of the WRN gene. Patient symptoms include premature wrinkling of the skin, graying of hair, cataracts, osteoporosis. It's most common in Japan, where it affects about 1 in 20,000, 1 in 40,000 individuals, somewhere in that range. Patients usually live into their 50s with the cause of death mostly attributed to cancer or myocardial infarction. Interestingly, mechanistically, Werner syndrome has been linked to all the hallmarks of aging, and there's more on that as well. In research conducted by Fang et al., NAD augmentation restored mitophagy and limited accelerated aging in Werner syndrome. NAD repletion restored NAD metabolic profiles and improved mitochondrial quality while extending the lifespan and delaying accelerated aging, including stem cell dysfunction. And this was in 2 invertebrate models, I believe, of WS. The figure A and B on the right are results from a C. elegans WS model demonstrating NR improvements to the function in embryo survival. Additionally, NR treatment had a greater effect on function compared to a CERP1 activator as well as a PARP inhibitor. And recently, a clinical trial to verify safety effectiveness of NR for patients with Werner syndrome was registered in Japan. The study currently enrolling patients at Chiba University in Japan will evaluate various safety and WS-related endpoints after 26 and 52 weeks of NR or placebo. We're looking forward to learning more about the outcomes over the next few years. It's going to take a few years before we get through that study. Citrin deficiency is actually one of the latest ones that we added this year. Citrin deficiency is a hereditary disorder, most common it's mostly an Asian-related disorder caused by mutations in the SLC25A13 gene that encodes for mitochondrial citrin transporter. Citrin is a component of the malate aspartate NADH shuttle and is essential for hepatic glycolysis. There are three types of citrin deficiency that manifest at different points of development. It's mostly in children ages 1 through 11, approximately. Symptoms include growth inhibition diagnosed as failure to thrive with dyslipidemia. There's also a form which is adult onset type 2, which causes ammonia and other toxic substances to accumulate in the blood. In adults, citrin deficiency can also result in neuropsychiatric symptoms, including nocturnal delirium, aggression, irritability, hyperactivity, delusions, and largely is provoked by sugar and or alcohol intake. In 2021, so earlier this year, ChromaDex developed a collaboration with the Citrin Foundation of Singapore to support the use of NR in citrin deficiency research. The first study by Paul Yen and other scientists at the Duke NUS Medical School in Singapore are going to examine the ability of NR to impact mitochondrial function, lipid accumulation, inflammation, and fibrosis in the liver using preclinical models that they've established for citrin deficiency. Last but not least, Duchenne muscular dystrophy, or DMD, is a disease characterized by progressive muscle wasting that is caused by a mutation in the dystrophin gene, resulting in dystrophin protein deficiency. By the age of 12, children with DMD are usually wheelchair bound. This leads to respiratory and cardiac failure, as well as muscle fragility, cellular dysfunction, impaired autophagy, calcium mishandling, oxidative stress. There's quite a bit going on here. DMD has been difficult to model in animals so far. In one mouse model of DMD, Ryu et al. In 2016 demonstrated that NR improved muscle function and heart pathology in dystrophin gene knockout mice. NR also improved NAD levels, improved oxygen consumption rate, and mitochondrial function in C. elegans models of DMD. In a different mouse model evaluated by the Selsby lab, multiple compounds, including NR, that were beneficial in the knockout model were not replicated in a newer, more severe form of this DMD mouse model. We have recently learned that there's an unpublished results from a case report that's currently under development, which will describe clinical benefits over 24 children with DMD following probably 5 or 6 months of NR supplementation. And we look forward to that also becoming published, hopefully sometime soon. So just a couple key takeaways is related to the work that we've been doing on the orphan side. DNA damage and mitochondrial dysfunction are associated with many orphan diseases and diseases of rapid aging. The etiology may include or be impacted by NAD deficiency. Preclinical models have demonstrated efficacy of oral NAD precursors on the progression of many of these diseases. And ongoing and new clinical studies are examining the potential benefits of supplementation with NR, or nicotinamide riboside. So, and I would one last slide that I wanted to leave you with. I've been talking a lot today about our CERP program throughout the presentation, which is largely an investigator-driven research program that ChromaDex maintains to advance the science behind NR, as well as other NAD precursors. NR isn't the only precursor that ChromaDex has, by the way. If you're interested in doing research on NAD precursors, please feel free to reach out to our CERP team. We're more than happy to evaluate or look at other potential collaborations as well. And Morten, with that, that's the end of my presentation.
Fantastic. Thank you. Thank you so much, Frank. Really great presentation. I enjoyed that you also referenced my old work in Will's lab. Really appreciate that. I was wondering if I may ask, with the CERP program, do you also provide financial support, or is it mostly on the material side?
It's both. We provide financial support in some cases. Without question, we'll provide the material for the study, whether it's powder that would be necessary for animal studies, or whether it's capsules with matching placebo, or in some cases financial support as well.
Okay, that sounds fantastic. We have a question on Slack from John Ferber. How does NR supplementation compare with nicotinic acid supplementation?
Well, they're different. Obviously, they plug in different ways. The problem we've seen mainly with nicotinic acid has been the side effect associated with high dose administration of nicotinic acid and the flushing effect that's caused by that. And that's one of the advantages of NR and NRK pathway, is that you don't have that. It alleviates that problem.
All right. Thank you very much. I think we will close the session here. Frank, it was great talk, and thank you so much.
Thanks, everyone.
And